still moving. Am I moving? Yeah, that's it. You know, one of the things you do is steal a professor a pointer and a clicker, and our life is miserable. Okay, so I usually carry 2 pointers just in case. So one of the things is, let's talk about today's lecture is the structure of the cell. And before I start, I'll tell you that I have a really cool task. And my task is in this block for you to teach cell biology. And cell biology is something you've been exposed to before, definitely. You wouldn't be here. So some of you took undergrad classes, some of you took grad classes. So some of the stuff we will cover in depth. Some we're less in depth that you have been covered. But one of the things you're going to notice is that our main focus will be to give it a clinical spin. So, you know, think of this when you think of preparing for exams or boards, is the questions will be kind of clinically relevant, you know. And I have put some clinical relevance here as much as I can, because you haven't had yet the entire biochem, you haven't had a farm, all these things. So I'm kind of just giving you a little taste of it. So don't do it as your previous year. One guy came to me and says, Dr. D, this is too easy. I said, that's great, you'll get A's, I get good reviews, we're all happy. However, it doesn't work that way, you know. So this is, so we'll start first with the structure of the cell. You guys know that we're all built by cells. It's kind of cool, think of this. We're built by around 100 trillion cells. All these little guys and gals are like gathering in different parts of your body, and what they do is they perform different functions. And by doing this, they make you survive everyday stresses. So the cells are the most minute living unit, okay, of your body. So that what makes them very unique is that they look like us, do they? This one looks like Dr. D. No, it doesn't. But what's a characteristic for the cell is this, is think of it, cells have their membrane, we have skin, okay? Cells have their organelles that helps them produce energy, do everything else, and we do the same. So look at the cell as a little human, because that's what they are in this situation. So the basic structure of the cell is we know that cell has nucleus, eureka. And you know, heard of the nucleus, and the characteristic of the nucleus is that it is surrounded by its own membrane, nuclear membrane. This membrane will extend and form endoplasmic reticulum, series of channels through the cell. And next is a cytoplasm, and cytoplasm is surrounded by its own membrane. It's known as cellular or plasma membrane. There is a term that you might have heard of or not. It's called protoplasm. What's a protoplasm? It's actually nucleus and cytoplasm take away membranes, no membranes, so the yucky fluidy thing, okay? So that actually is a protoplasm. So what are the structures that form protoplasm? Water. We're mainly made of water. 80 to 90% of the cellular structure is water. Then we put a little spice in it. There are some ions in it. There are proteins in it. Very important. Lipids and carbohydrates. Specifically in tomorrow's lecture, we're going to talk about membrane that has a lot of lipids and carbohydrates in it. So that's where you're going to find it. But there are lipids and carbohydrates also in cytosol. Now let's talk about organelles. What does it mean, organelle? Organelle means a cute Latin word for little organs. Okay? So what are the organelles? Organelles are little organs inside a cell. They're surrounded by membranes that have the same structure as cellular membrane. And you'll find them in cytoplasm. And what's important, what I always thought when I was in undergrad, is that organelles kind of float around the cytoplasm. No, they don't. They're actually fixed in different locations by the presence of cytoskeleton. Cells have their own skeleton. We're going to talk about that. And this cytoskeleton anchors into the organelles and keep them in a specific spot. It can move them. It can contract and move the organelles in different directions through the cells during different functions. So that is a characteristic of it. The first one we're going to talk about is nucleus. So nucleus is the largest subcellular organelle. What does that mean is whenever you look at the cell, you might see one or several nuclei. And that is very important because it houses genetic material. It's involved in the process, that's not only function, but major function is houses genetic material for replication of the cell. Now, one of the pearls of wisdom for today is that all eukaryotic cells have nucleus. Not correct. Correction is that red blood cells actually don't have nucleus. Red blood cells, the cells that carry oxygen through your body, don't have nucleus. Why? Because they don't need to replicate. They really are just a vessel that carries oxygen through your body. So red blood cells are formed inside the bone marrow, and you're going to hear from me later about that. And they will have nucleus. Eventually, they will spit out their nucleus, get rid of it, and become mature red blood cells that will carry oxygen throughout the blood and help you breathe. Now, nucleus contains chromosomes. We know which are primary genetic material. They contain enzymes, very important, that are involved in transcription of chromosomal DNA. They also contain enzymes that are involved in the production of RNA, which means is a production of protein that is very important in the function of the cell. They also have transcription and chromatin remodeling factors that actually regulate the transcription of RNA and regulate the process of production of protein further down the road. Nucleus is surrounded by double membrane. Actually, one of my colleagues who was doing PhD with me did a huge 10-year study on specific nuclear pores. So, nucleus has its own membrane, and that this membrane is continuation of endoplasmic reticulum membrane. And this membrane of nucleus have holes or pores, which are known as nuclear pore complexes. Why I wanted to tell you the story about my friend, he spent 10 years at science of working on a specific nuclear pore, and during his defense, I actually asked him, why is this important? And his response was, don't know. So, welcome to science, okay? 10 years of work, but that's okay. He's gonna have a pore named by him. So that's kind of cool. So the pores are actually very small in a nucleus. There is 16 to 60,000 Daltons as their size. What does that mean? Does that mean that the protein larger than 60,000 Daltons cannot pass through this pore? No. Protein can fold on itself, can squeeze, change conformation, and go through it. Now, these pores are very important because they enable communication between nucleus and cytoplasm. They enable the communication between these two. Now, welcome to medical school lecture. So we're going to mention one condition that actually affects the nuclear membrane. You're going to hear more of them from Dr. Traore in the future and Dr. Lucia. But right now, I'm going to mention one condition known as Emery-Dreifuss muscular dystrophy. Rare condition. Rare is the condition, more common it is on your board exam. So this is a very rare condition common on your board exam. Okay. So it's actually a disease of nuclear envelope, and it affects individuals through X-linked dominant inheritance. What does that mean, guys? X-linked dominant, which means both males and females will get it, because it's X-linked dominant, because males will get it because XY, females will get it because they're XX, but they have a dominant gene, so they will actually get the disease. It's rarely recessive. It's characterized by a mutation of EMD and LMNA genes, which are known as, which produce proteins known as emerin and laminin. Look where they are. This is the membrane of the nucleus, and you see here emerin and laminins. And they're very important. Why? Because they provide connection between the skeleton inside the nucleus and skeleton outside the nucleus. So they almost anchor the skeleton inside the cell, inside the cytoplasm. So what's going to happen when these proteins are abnormal in these individuals? They will disrupt nuclear and cytoplasmic communication. So there's going to be a problem with communication between nucleus and cytoplasm, and it will affect specifically striated muscles. Specific group of striated muscle affected will be heart. It's a type of striated muscle. It's a cardiac muscle. You're going to hear a lot from them in Block 2 from me as a part of the cardiovascular phys. But what is going to happen is these individuals develop dilated cardiomyopathy. The heart will be much enlarged. It will not be able to contract well. And they will develop limb girdle muscular dystrophy. And that's characteristic that these individuals have weakness that will start from the end of their extremities, arms and legs, and will start moving toward the center. And that is known as a limb girdle muscular dystrophy. They will develop contractures, and they will develop slowly progressive muscle weakness and wasting. A few interesting videos that you can kind of click on later if you want to look at this disease a little bit more. The next is nucleolus. What is nucleolus? Inside the nucleus, it's a cloud. Kind of cool. Why? Because it is a dense material inside the nucleus. It doesn't have its own membrane. Okay? So there can be one or more present in nucleus, and we see them here. And what's a characteristic of it? Again, that it does not have a membrane, but it is a site of transcription for genes that encode for rRNA, ribosomal ribonucleotide acid. So the large and small ribosomes are fully assembled inside the nucleolus, and they're transported then into cytoplasm to produce protein. Now, they're found enlarged. So you will see these nucleoli large in cells that produce a lot of protein. Why? Because why? They need to produce more ribosomal RNA. They need to produce more ribosomes. More ribosomes produce more protein. So you will see enlarged nucleoli in malignant cells. Okay? Cells that are producing a lot of protein, they're growing abnormally. Okay? So again, remember that malignant cells produce large amount of ribosomes and specifically protein. Here is a ribosome. Is it a funny looking fellow? Look, it's like a mushroom. Come on, there's a head on top and a little thing on bottom. But ribosomes, I compare to a machinery in our cells, our factory production, okay, of proteins. Cells will grow and survive because they produce proteins. So they're cellular machinery for protein synthesis. And they can be free floating. They can be really cool, free, yay, traveling through the cell. Or they can actually be attached to endoplasmic reticulum and a part of the system. They work for the man, okay? So what are they? They're attached to endoplasmic reticulum where they produce proteins next to the endoplasmic reticulum. Their size is ATS. What does it mean, ATS? Dr. D's weight is ATS. No, that's not how it is. ATS is actually the measurement of how you isolate them through centrifuge speed. So don't get confused. It's not a physical measurement of weight or a size. So this size is ATS. And ribosomes have two subunits. They have a large subunit and a small subunit. Large subunit is 60S, and a small subunit is 40S. So you see, 60 plus 40 will give you 100. So that's not the same value. This is just a value of how do you isolate them using certain speeds of centrifuge. So the large subunit has 3 RNA molecules, 5S, 5.8S, and 28S, which they usually test you on boards asking you this. Less and less, thank God. And 49 proteins. While a small subunit, the 40S, has 1 rRNA molecule and 33 proteins. And there is really 2 cool videos, guys, if you want to look on your own, kind of explains how they work and how the ribosomes work. I'm going to touch upon the role of ribosomes. We all know that ribosomes are involved in the production of protein. Okay? So how this process of translation of genetic code into the protein sequence, into the amino acid sequence in a protein. So that's where the main role is. So we know that 40S subunit will first bind to the inducer tRNA. So this is the first tRNA that will carry amino acid on its top. So the small unit will first bind to methyl transfer RNA. It's called inducer tRNA. And will also bind to the mRNA on the fifth end of mRNA molecule to the corresponding codon that is known as a start codon. Now, the small unit will now recruit the large unit of ribosome. So the large unit will attach and form sandwich here, can attach on top of this complex. Now the large subunit will now read this complex, actually, will read the next codon, next triplet, which will now be interpreted by the large unit. And the large unit will now bring in corresponding tRNA that has corresponding anticodon. Now, tRNA with corresponding anticodon will bind to the second spot. And now the complex will stimulate the formation of peptide bonds between two amino acids that are carried of tRNA. Now, the first tRNA will be removed. In this situation, it's the initiator tRNA. And what's going to be left is a second tRNA with these two amino acids attached to it, connected with the peptide bond. And this second one will now be moved further into the first spot, and the next codon will be read, and the next tRNA will be attached. I know you heard of this before. That's why I'm going superficially through this, because this is actually the process of translation. You will hear it again from Dr. Quo, I think, coming up, or actually you'll hear it from Dr. Lucia coming up. So as this happens is, look, you see here ribosomes, and you see growing protein chains produced from those ribosomes, and they will eventually, when the stop codon is reached, they will be detached and they will form proteins. So why is all this important? This is important for survival of us and our first cousins, bacteria. Okay? But there is a catch. The ribosomes of bacteria are built slightly different than ours. So ribosomes of bacteria or prokaryotes are 70S, not 80S, and they consist of 50S large unit and 30S small unit. So why is this important? We can target it. If we want to kill bacteria, we will target the ribosomes. Why? Because the ribosomes are different than ours. And if we stop the protein synthesis in the bacteria, we will actually stop the growth of bacteria and will kill bacteria. So that is the way how some antibiotics work. So just an example, chloramphenicol will bind to a 50 portion and will inhibit the formation of peptide bond in bacteria between two adjacent amino acids that are still attached to tRNA. Erythromycin will bind to 50S portion and will prevent the translocation and movement of ribosomes along the mRNA. It will block and almost stop the movement of ribosomes down the mRNA. Tetracyclines is another group that will now interfere with 30S portion, and they will interfere with the attachment of tRNA to the ribosomal complex. And streptomycin, and you're going to hear more in details in this, will change actually the shape of 30S portion and will now cause the code to be read incorrectly from the mRNA, and abnormal proteins will be produced in bacteria, and bacteria will die. So this is actually, you see the connection between the basic science and the medicine that you're going to apply in the future. Let's talk about rough endoplasmic reticulum. It's actually assembly line of our cells, and it is a teenager with a lot of pimples. Okay? The best way to explain it, it's actually internal membrane that is continuous with the outer membrane of the nucleus, and this membrane is studded with ribosomes. And what is the importance of the rough endoplasmic reticulum is a production of protein. Those ribosomes are transcribing all those proteins, and all those proteins now are going to be stored in the rough endoplasmic reticulum. So they're very much important in production of membrane or secretory proteins, and we have to understand the proteins that are produced here on ribosomes that are attached to the rough endoplasmic reticulum will have specific amino terminal sequence. They will be labeled. They will be having label at the end of those proteins that will tell your cell what they will be used for. Okay? So I might have a label of, I'll be lysosomal protein Y because I like to fight. So I'm going to be going into the fight forming lysosome. So these sequences of these proteins that are produced on those ribosomes are recognized by ribosome-associated particle. And this protein, ribosome-associated particle, will shuffle proteins into the specific docking sites on ribosomes and endoplasmic reticulum. Now, this protein that has been attached to ribosomal-associated particle and ribosome and endoplasmic reticulum will be now put and pushed into the rough endoplasmic reticulum. Now, these proteins will be inside the rough endoplasmic reticulum after they've been produced, and they will become glycosylated. They will be added a carbohydrate, and they will travel inside the endoplasmic reticulum. And these proteins will now be traveling through the rough endoplasmic reticulum all the way to the area known as smooth endoplasmic reticulum, where they're going to be then utilized. So you see this, proteins are formed on ribosomes. They have the targeting or labeling sequence on it. These proteins are now attached to the ribosome-associated particle. Ribosome-associated particle will now help together with ribosome to put these proteins into rough endoplasmic reticulum. These proteins inside the rough endoplasmic reticulum will get glycosylated, become carbohydrate attached, and now they will travel through the rough endoplasmic reticulum all the way to the smooth endoplasmic reticulum, which is also known as transitional endoplasmic reticulum. So what's going to happen when these proteins are in a smooth endoplasmic reticulum? They will bud off of the smooth endoplasmic reticulum, and they will actually now travel all the way to another organelle known as Golgi apparatus. It looks like flattened sacs. What's a Golgi apparatus? The best way to explain Golgi apparatus is it is a storage place. It's storage place where shipping happens in the company. So the transport vehicles that are detached from the smooth endoplasmic reticulum will now fuse with cis end of the Golgi apparatus. Now they will be transported through cis end. Cis end is the end towards the nucleus. So now they will be transported through medial end and the trans end of the Golgi apparatus. And inside, these proteins will be modified, okay? So they will be modified and processed and matured. And now they will pop at the surface of the trans end of the Golgi apparatus that is facing the membrane. And they will now bud off of the trans end. You see being surrounded by the same membrane as Golgi apparatus. And they will form secretory vesicles. And these secretory vesicles will now travel to a predetermined site in the cells where they're going to be utilized. One of the important things is, just a pearl of wisdom, is if you look at neurons in patients with Alzheimer's disease, you will actually see fragmented Golgi apparatus. So we think that with patients with Alzheimer's disease and dementia, there's a problem of transport and maturation of these proteins through the cells that are produced in neurons. Now, smooth endoplasmic reticulum is actually continuous extension of rough endoplasmic reticulum. And what is the characteristic of smooth endoplasmic reticulum? We mentioned it. It will carry all those proteins. They will be then pop out of it and travel to Golgi apparatus. But another important thing is that smooth endoplasmic reticulum doesn't have attached ribosomes on it. And more importantly, it's involved in lipid metabolism. That's the area where actually the lipids are broken down in a cell, and they are location of final production of detoxifying enzymes that help detoxify your cells. One of the most important functions of the smooth endoplasmic reticulum is storage of calcium. Smooth endoplasmic reticulum, and you're going to hate Dr. D because I'm going to be talking about these when we talk about muscle fizz in next block. A smooth endoplasmic reticulum, specifically in cardiac cells or other cells, is actually a storage for calcium. That's where most of the cellular calcium is held in a smooth endoplasmic reticulum. So if you stimulate the cells through a certain stimuli, you will actually cause a formation of the secondary messenger called inositol 3-phosphate, which we're going to talk about in upcoming lecture. This inositol 3-phosphate will now stimulate the smooth endoplasmic reticulum to release calcium. So this calcium will go out of smooth endoplasmic reticulum into the cytoplasm. And this calcium will activate a number of cellular pathways and will activate certain processes such as in cardiac cells contraction. So release of calcium from smooth endoplasmic reticulum will stimulate the contraction of cardiac muscle. It's one of the things. And now, importantly, is that the surface of smooth endoplasmic reticulum contains a lot of calcium pumps. What does that mean? That means that that calcium that is released from smooth endoplasmic reticulum will eventually pump back into the smooth endoplasmic reticulum at the end of the signal, at the end of the contraction, for example, in cardiac myocytes. So that is one of the characteristics of the smooth endoplasmic reticulum. It acts as a sponge for calcium. It holds the calcium, releases it, stimulates it, and puts it back in. Clathrin-coated pits. These are structures that I really like because they have clathrin-coated pits have actually very cool name. But what their function is, look, they act as baskets that will carry the content from the surface of the cells into the cells to be digested. So clathrin-coated pits, a good example, you remember those machines that kind of grab and they never work and you always take your money? That's what the clathrin-coated pits are. So they're dimple-like structures on the cell membrane, and they contain receptors for specific extracellular compounds that cell wants to eat, okay? So what they're going to do is they actually have this hemibasket protein structure on the bottom of the pit, okay? So this is clathrin. Clathrin protein, as you see on the bottom, will kind of form a basket, okay? So moment when the compound attaches to its receptor that the cell wants to eat, the clathrin will change its structure and it will encircle the compound, okay? Almost the basket will close around the compound, okay? And now this compound, this material that's been eaten by the cells will be pushed into the cell. Okay? All in that basket. So clathrin-coated pits are involved in the receptor-mediated endocytosis, uptake of compounds outside the cells into the cells. And clathrin will now polymerize the moment that the compound attaches to its receptor, and it will close around the compound and will act as almost now as a cage that will push this compound into the cell. Now, as the clathrin is taken into the cells and this basket is taken into the cells, this clathrin will be depolymerized. What cell is going to do is going to eat away the basket. It's going to destroy the basket, okay? And the next thing is it's going to acidify the content that was in that basket, adding acid in it. And this will result in formation of early endosomes. Now this content will be more acidified and more acidified inside, and will now become, and the receptors that were inside the basket that the compound attached to will return to the surface. Now these early endosomes, these vesicles that are containing the compound that the cell ate, will fuse to each other, forming a late endosomes. And these late endosomes will have very low pH in them, and these late endosomes will now be subjected to lysosomes. As you remember, and lysosomes are the organelles that will eat away the content. They will destroy the content. Another transport protein I want to mention is caveoli, or protein is known as caveolin. And the characteristic for caveoli is it works very similar as clathrin, but it does not destroy the compound. Caveoli will attach to the compound. They will take the compound and transport it all the way through the cell, and then release it on the other side of the cell. So these caveoli are involved in transcytosis, a transport of compounds through the cell, not into the cells and to be eaten away, but through the cells. And they have actually very high concentration of growth hormone receptors, so they're involved in transport of hormone. Lysosomes. Lysosomes are awesome. Why? Because they're the digestive system of the cells. So lysosomes are membrane-enclosed acidic compartments, and we see here lysosomes in the cell. And they will contain huge amount of lysosomal enzymes, over 40 enzymes. And these lysosomal enzymes are known as hydrolytic enzymes. Do you know what hydrolysis means? What does it mean? You take away water. You're right. from anything else, and as you do, you break down compounds, okay? So that is the characteristic of it. So lysosomes eat away the foreign material. They eat away the foreign material. They eat stuff that the cell has endocytosed. But importantly is that these hydrolytic enzymes that are inside lysosomes require a very acidic environment. So that's very important, that lysosomes have very high, very acidic pH. Now, hydrolysis, these enzymes are produced in rough endoplasmic reticulum. As you remember, they are proteins, so they're transported into Golgi apparatus. In Golgi apparatus, they're added a mannose 6-phosphate tag that will tag these proteins as destructive. That these hydrolysis tags adds, this tag will tell your cell that these are bad boys, okay? So what this tag, 6-mannose tag, will now make Golgi apparatus put them, pack them, and excrete them as lysosomes, okay? So, because these are the bad boys. So what is their function? They will degrade endocytosed material that cells eat, but they will also eat away phagocytosed material. And more important, they will eat the cells itself. What does that mean? Can you imagine? That will be awesome. I have a large nose, for example, and then I want to get rid of that nose. So I choose to send lysosomes there, destroy it, and grow a new one. Okay? So that's what cell does every day. That's why we survive. It's constantly repairing itself. So these lysosomes are not only destructive or involved in digestion, but in constant repair of the cell. Now, everything that is broken down in these lysosomes, such as amino acids, sugars, and nucleotides, are transported out of the lysosomes into the cytosol for reuse. So what happens is, that means lysosomes will break down that compound, and there are some yummy amino acids in it, okay? There are some sugars and nucleotides. Oh, sugars, how much I hate you. But, so what happens is that these one are be sent into the cytosol and will be utilized. Now let's touch upon a little bit of clinic. Lysosomal storage disease. Oh, how much you will hate them. Okay. Why? Because I'm just giving you 3. There is probably 12 that they ask you on boards. Again, what did Dr. D teach you? Rarer disease, more common it is on boards. Okay, so these are rather rare diseases, but they'll test you then on boards. So I'm going to mention only 3. So what are lysosomal storage disease? What's going to happen, guys, if your body is producing abnormal hydrolytic enzyme? 1 out of these 40. What's going to happen when that compound is digested? You have abnormal lysosomal enzyme. It's not going to be digested. So what's going to happen with that compound? It's going to stick around, and it's going to build up, and cell become fatter, and eventually it will die. Okay, so that is a lysosomal storage disease. Means that lysosomes are stored in a cell. Why? Because they can break down certain compounds. So I'm going to mention, so indigestible material will impair normal cell function because they're missing a specific hydrolytic enzyme, or hydrolytic enzyme is abnormal. So I'm going to mention 3. Tay-Sachs disease is characterized by congenital absence of lysosomal enzyme hexosaminidase. Awesome name. Can you say it fast 6 times? Hexosaminidase A, hexosaminidase A. I tried it yesterday. But, so this enzyme, these individuals, Tay-Sachs disease, this is a congenital disease, is an hereditary disease. It's actually, they don't have hexosaminidase A. They don't produce a normal hexosaminidase A. So this will result in accumulation in their cells of GM2 ganglioside, which will lead to neurodegeneration because it's going to affect neuronal cells. It will lead to developmental delay. Cherry red spots on the back of the eyes are one of the characteristics. Lysosomes will have onion skin appearance. If you look them in the microscope, specific electron microscope, you'll see these lysosomes that have layer after layer after layer of deposits, okay? So it looks like an onion. And they will not have hepatosplenomegaly, and I'll explain you why. They will not have enlarged liver or spleen. The next is Gaucher disease, which lacks glucocerebrosidase. So these individuals will accumulate glucocerebrosides inside their cells. They will have pancytopenia, which means most of the cells in the blood numbers will be decreased. They will not produce normal blood cells. They will have aseptic necrosis of the femur. Their femur will break easy. They'll have bone crisis. They will have Gaucher cells, which are these macrophages full with undigested material, okay? And they will have hepatosplenomegaly. They will have enlarged liver and spleen. And the next is Niemann-Pick disease, which is characterized by the absence of sphingomyelinase. You know what myelin is? It's the coating that coats neurons. So this, they can't digest myelin. So these cells will have lack of sphingomyelinase, where they will accumulate sphingomyelin. So they will develop what? What system will be first affected? Myelin. Nerves. So these individuals will develop neurodegeneration. They will have foam cells that are very similar to Gaucher cells, that are these macrophages full with undigested material. They will have cherry red spots on macula, same as Tay-Sachs disease, and they will have hepatosplenomegaly. So let's look clinically, guys. So if you have a patient with similar, look at this, Tay-Sachs disease, neurodegeneration, is it? Niemann-Pick disease, neurodegeneration, okay? So these ones have foam cells, the Tay-Sachs cells don't have. But they both have cherry red spot of macula, but one has enlarged liver and spleen, other one does not. That's how you learn it. You compare things, okay? And as you compare it, you will be able to identify it and diagnose. Now, next thing is, this is a cherry red spot on macula in Tay-Sachs and Niemann-Pick. This, what you see, is an onion skin lysosome. Looks like an onion skin in lysosome. It's a Gaucher cell, which is a macrophage full of this lipid foamy material that was not digested. It's a foam cell in Niemann-Pick disease. And these are just the basic characteristics of the Gaucher disease. There are different levels. There's Gaucher 1, 2, and 3. And you're going to hear more about this in your biochem, way more than you hear from me. And I wanted to show you that connection. Peroxisomes, my favorite. Actually, these are my favorite organelles. Why? Come on, guys. Something is called peroxisome. Nobody's getting it. Okay, it doesn't matter. It's kind of awesome. Why? Because you could pack it and sell it, peroxisomes for sale. You know, think about it. You can't say Golgi apparatus for sale. Who cares? But peroxisomes, that's important. Now, why I'm telling you this? I spent a lot of my research days on these peroxisomes, so that's why I like them, actually. But peroxisomes are very important. Why? Because I worked on them, number one. Number two, because since I worked on them, I might test you on it. Come on, that's how you're going to learn in the med school, because, oh, Dr. D likes his peroxisome. So, this is it. But what's the function is? They're involved in breakdown of cellular lipids, and they are involved in beta oxidation of long-chain fatty acids from membrane lipids. They actually break down and help rejuvenate membrane. Come on, think about it. Rejuvenate membrane, rejuvenate your skin, okay? So that's what I wanted to tell you. So this is interesting is, so they're important. They also produce acetyl molecules from breaking down long-chain fatty acids. And the molecules that are produced by them are transported into the cytosol and used by the cell for any kind of reaction. Most importantly, my research is that peroxisome contain an enzyme known as catalase. So what's the characteristic? Your cells in body are producing hydrogen peroxide. Do you think, guys, hydrogen peroxide is good? No, you can put, if you all pass this exam, I'm going to use hydrogen peroxide on my head and I'm going to look white for a while. And my wife would kill me. Why am I betting with this? Because I know that's not going to happen. But I'm just joking. Yes, it will. It will. So I didn't say this. Nah. My big mouth puts me in a lot of trouble. But that's what my wife says all the time. But this is it. But peroxisome contains enzyme catalase, and this enzyme breaks down peroxide. Look at this. So it actually makes and what is associated with aging is increased production of hydrogen peroxide. Hydrogen peroxide will destroy your cells. Older you get, less you can quench that hydrogen peroxide. So this is a rejuvenation enzyme, okay? So catalase will utilize this hydrogen peroxide and break it down into water. And water is not bad for us, so we can actually take water. But also these peroxisomes are important in synthesis of plasmalogen, which is a very important compound it's a phospholipid that is found in myelin and in heart cells. So it's very, very important. And defect in peroxisome function will lead to X-linked adrenoleukodystrophy and Zellweger syndrome. So Dr. D is going to talk about Zellweger syndrome. Why? Because of the actress, okay? And why is this? This is, again, a very rare condition, however common on boards. It's characterized by abnormal production of peroxisomes. So these individuals will develop something called cerebrohepatorenal syndrome. What does that tell you? What is going to be affected in these individuals? You see, medicine is straightforward. We just like to call it Zellweger syndrome so patients will not know what we're talking about. But what it is? Cerebral, brain, hepato, liver, renal, kidneys. Come on, man. You know, if we called it this way, we will lose jobs. So what we do is we call it Zellweger syndrome. Okay? So one of the characteristics for this is this. It's the most prominent manifestation of Zellweger spectrum of disorders. So think about it. There is actually a condition. This is a spectrum. There is a Zellweger syndrome, 1, 2, 3, 4, 5. Dr. D doesn't want you to know this. I love addressing myself in the third person for some reason for a while. But that's cool. But listen to this. So these individuals will not have functional peroxisomes inside the cells. It is actually autosomal recessive disorder caused by mutations of the genes that encode for peroxins. Peroxins are proteins that form peroxisomes. Okay? So these individuals cannot form peroxisomes. They will have impaired neuronal migration. They will have hypomyelination. They will not produce myelin appropriately. What's going to happen when you do not isolate neurons? You will have signals sent from neurons in different directions. Okay? Because myelin is an isolating sheet on neurons. So when you don't have that, if one neuron fires, it will easily stimulate other neurons. Okay? So these individuals will develop seizures because of that problem with neurons. They will develop cranial deformities, problems with hearing and vision. They will develop chondrodysplasia punctata, which means is that certain portions of their bones will turn into cartilage. Okay? And they will develop cysts in their kidneys, apnea, they will have problems with breathing. And sadly, individuals diagnosed with this disorder usually don't live beyond age of 6 months. Now, mitochondria are power houses of our cells. That's where we produce ATP. ATP is green stuff, okay? ATP is energy currency in our body. The entire cell works utilizing that good old ATP. So who makes ATP? Is that ATP is actually made by mitochondria. So you see how it looks like. What's so cool about mitochondria? It has envelope that contains two membranes, inner and outer membrane, okay? Mitochondria we have inherited from bacteria. Think about this. So what does that mean? Is at one point our cells have chosen to steal mitochondria from bacteria, and we use it all the time to produce energy. Why is this? It's because the DNA inside mitochondria is different than the DNA outside in your cell. So this is called a circular DNA, and also they have their own ribosomes that produce their own proteins inside. So almost act like a cell inside a cell. Mitochondrial DNA is from whom? From your mom. So this is a geeky way of medical student / doctor sending card to his mom. I did that. I said, Mom, thank you for mitochondria. So my mom then told my dad, so you thought I was not important. I said, my dad, no. You see, you gave me mitochondria. So, but what it is actually if you look for lineages, you look for this DNA inside mitochondria, and you can follow maternal line all the way to however you want to go back. Okay? So these are inherited directly from mother. This DNA inside and the way that ribosome produce proteins. They have folded membranous structures called cristae, and inside the membrane is covered by oxidative enzymes, enzymes that use oxygen, known as cytochrome oxidase. We see that cristae will protrude into mitochondrial lumen, into mitochondrial matrix, and this matrix, this gooey stuff inside, is actually involved in extraction of energy from nutrients. So this is how what they do. Inside the mitochondria, citric acid cycle reaction will occur, which will produce energy, will break down food, it will digest food. So the citric acid cycle will take place inside the mitochondria, inside the matrix. It will produce NADH and NADPH, nicotine adenine dinucleotide. And now the electrons produced by this process will be used to run pumps. Guys, I'm just giving you a brief overview. You will hear all this in details, and you will hate Dr. Traore and not me, okay? But this is it. So these electrons that are produced by citric acid secretion are used to run pumps that are on the surface of the mitochondrial membrane. These pumps will push protons, hydrogen molecules, from inside of the mitochondria into the space between two membranes. Make sense? And now these protons, this hydrogen concentration in between these membranes will become really, really high. So where these protons want to go? From high concentration to low concentration. So they will want to go from the space between the membranes back into the mitochondrial matrix, across the membrane. And they will be let go through that membrane through a little pump called ATP synthase. ATP synthase. And ATP synthase will use that energy of these protons going back from the space between the membrane into the matrix, okay? And they will use that to produce ATP. Just to show you, here it is. So this is the membrane. This is inner membrane. This is a space between two membranes above. Look, electrons produced by citric acid cycle are used to run the pumps. You see these pumps. These are known as complex 1, complex 3, complex 2, and complex 4. These electrons will run the pumps, and these pumps will pump hydrogen ions from inside of the mitochondria into the space between the membranes. So what's going to happen in the space between the membranes with these protons? There's going to be a lot of them. Hydrogen ions are going to be a lot in that space. So they will like to go back into the matrix. So as they go back, they will run the ATPase pump that will produce ATP. And this is how we make energy. This is how we produce energy. Cyanide poisoning is very common, specifically after exams on the side of faculty. But cyanide poisoning, when Hitler killed himself, but he was a coward, so he had to shoot himself in the meantime. But what it is that cyanide poisoning is actually, as you heard, is like, I don't know about you guys. My wife likes to fall asleep with killer stuff. So if I don't show up on Tuesday or Wednesday, she falls asleep watching all these shows, about who killed whom or whatever. And, and when you sleep next to it, I said there has to be some subconscious influence on her. That's going to eventually, I will croak here. So I'm just joking. No, don't worry. She's a psychiatrist. She wouldn't kill me. She would analyze me. But the thing is, but it's used in sophisticated suicides. The more common it is, cyanide is produced by actually burning of some synthetic fibers. Actually, cyanide is used as fire retardant. Stupid. Why? Because when it burns, if it burns, it will release, it will be used to put it on the surface of blankets, okay? So what it's going to do is going to suppress fire. But when it burns, it will release cyanide, okay? So this cyanide is a common problem in house fires. Cyanide will attach to cytochrome A3 subunit. Remember those pumps we talked about that carry electrons? So one of them will be blocked by cyanide. So they will not carry, they will not be running. When the pumps are not running, what's happening with protons? They do not get pushed into that space in between the membranes. So they don't come back. So they don't produce ATP. So suddenly cell starts producing energy. The person dies. No energy, no ATP, person dies. So that is actually the mechanism of cyanide poisoning. Now, cyanide poisoning, one of the big things, if you remember Jonestown Massacre, where people were drinking that cyanide-laced Kool-Aid. But what's a characteristic, and thousands of people actually died. But more common in ER, you will see smoke inhalation associated with cyanide poisoning. Okay, we mentioned why, because it's used as retardant, fire retardant, typically. But what happens is that it's less and less. But that cyanide gets released during burning, so these are different levels of the effect that they will have. And the therapy is amyl nitrate, sodium nitrate, and sodium thiosulfate. Why I'm giving you this? It's just for fun of it, because it makes me look smart. But the thing is, truly, that's a board question, is these are going to be give patient after they get inhaled that cyanide. Why? Because they actually change chemical composition of cyanide. They convert it into thiocyanide that is white like toxic substance. These are my lecture objectives, and you have some clinical cases at the end that you can go on your own. It kind of shows you the way you think as you progress through this. But before I leave and before anybody else shows, I kind of want to share something with you. Let me see if I can find it.